matrix of the mitochondria of all our cells is the cycle of reactions that won Hans Krebs a Nobel Prize. The cycle takes the energy in food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. And Krebs makes FADH2, its function is the same. Krebs also makes some ATP, another claim to fame. We're talking Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes one ATP, 3 NADH, 1 FADH2. Right before the cycle's a transitional part, links Krebs to glycolysis so Krebs can start. Enzymes break a CO2 off a pyruvate, yields an NADH and acetyl-CoA. Acetyl-CoA carries carbons too, with all the energy that derives from food. It's a highly reduced molecule with energized electrons that fuels up the Krebs cycle as it cycles on. Now at the start of Krebs, this acetyl-CoA has the 2 carbons it carries in ripped away. Enzymes put these on oxaloacetate with carbons 4, make 6 carbon citric acid. Who could ask for more? Notice 3 carboxyl groups on the citric acid. That's why the cycle's also named for tricarboxylic acid, or TCA cycle if it's acronyms that you prefer. Krebs in honor of its discoverer, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Enzymes work on citric acid and remove the CO2, and other enzymes modify and oxidize it too. The 5-carbon result is alpha-ketoglutarate. Krebs cycle, it's so great. Every oxidation can power the reduction of NAD+, which gains electron carrying function, become an NADH, that energy sensation, which later on in respiration powers ATP creation. Another oxidation follows another CO2 remove, leaving us with a 4-carbon molecule. Another NADH results from this oxidation. As Krebs does its energy transformations, this 4-carbon molecule, succinyl-CoA, still has lots of energy enzymes can take away. A series of reactions yield 1 ATP, the cell's main energy currency, leaving just enough energy for enzymes to reduce an FADH to FADH2, and one last NAD+ will also get reduced as the final electron carrier NADH gets produced. We've harvested what energy came in at Krebs' start. Now we have oxaloacetate at this final part. Oxaloacetate is the commencement and finale, ready to meet acetyl-CoA and here at the final tally. Krebs goes round and round, such an ancient cycle, spinning like the wheels of my bicycle. Krebs like the axle of aerobic respiration. I breathe out its CO2 with every exhalation. We've walked through the cycle, so now let's review. Input is acetyl-CoA with carbons too. The carbons get removed, releasing CO2. Exhaling sends the CO2 out of you. The cycle's functions: energy transformation, 3 NADH, 1 FADH2 creation, and also synthesis of 1 ATP, which cells directly utilize for energy. For every glucose cells absorbed, the cycle runs 2 times. As long as cells get fuel, Krebs is running just fine. It precedes electron transport chain, it follows glycolysis. It's spinning round and round in the mitochondrial matrix, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2.